next paper is bacterial load of virtual reality headsets. And this is another live presentation. Yeah, this is another live presentation by G. Adam Jones from the University of Mississippi. So yeah.
All right. Hello, everybody. I'm Adam Jones with the High Fidelity Virtual Environments Lab, the High Five Lab at the University of Mississippi. And today I want to virtually talk to you about some research that we did that's interesting. You'll see what I mean. So the title of our paper is Bacterial Load of Virtual Reality Headsets. This is work primarily done by Benjamin Creel, who since graduated and gone off to med school and is starting to do wonderful things. Caitlin Renz-Jones, a talented geneticist, also my wife. Our colleague Colin Jackson heads the Jackson Microbiome Lab here at the University of Mississippi, and myself. So with a title like this, what do you think we're going to talk about today? Well, you're either not going to be surprised or be surprised. We'll see. Anyway, so virtual reality, we know it, we love it. Most of you have probably been working in it for a long time, but the environment that VR has been evolving in has been changing. It used to be a large, bulky, expensive technology, and the number of users per device stayed fairly low. But times have changed. VR displays are getting much more affordable, meaning they're much more accessible, meaning they're making it into wider places. And I suspect a lot of you are like me, and you're working and researching in probably a university setting. And in these settings, you've got people who are putting virtual reality lab spaces all over the place. You've got them showing up in libraries. You've got them showing up in learning enhancement centers. You've got them showing up in classrooms. And a lot of you are probably like me and also offering classes in virtual reality development. Now, the problem here is that with any open lab space where you have people coming in and sharing technology, there's going to be the potential for contamination of the technology with pathogens. And virtual reality is a weird technology in that it's an intimate technology. It's a technology that we literally wear on our faces. It docks with us. It communicates data through us through a biological data port, our eyes and ears. And this can offer a unique vector for potential pathogen contamination of the display and subsequent users. So in our class, my observation has been that students don't like to observe sanitation practices. And this can be potentially detrimental, especially when there's the potential to spread pathogens to other users. In our class, we actually, so flipping to our next slide, so in our class, we primarily use the HTC Vive. And because, as a lot of you probably noticed, people sweat when they move around in virtual reality a good bit. And this can cause the stock sort of foam padding face pads on these displays to become saturated with sweat. So one of the first things we do is we actually swap those out for sweat-proof pads. This hypothetically makes cleaning the displays easier, less cumbersome. And the students are versed in how we want them to sanitize the displays in our virtual reality class up front. But the observation that I've made and that my grad students have made is that people don't typically follow your sanitation procedures. Excuse me. So one of the questions I wanted to ask was, how dirty do these displays get? And so we partnered with our colleagues in the biology department to do a brief study during the spring 2019 semester, so concluded almost a year ago now. And the procedure was the students in the class were versed on sanitation procedures, instructed to do them. Our graduate students do a forced sanitation of the displays once weekly anyway. And once a week, we had a student from biology come in and swab the displays. And specifically, the displays were swabbed in these two areas, the areas in the red boxes there. We did a swab across the forehead and a swab across the nose pieces of the displays. And then these samples were then analyzed for bacterial contamination. Now, in some ways, with having me give this talk, you got the short end of the stick because I'm the only non-biologist on the paper. But then again, maybe it's a good thing because, there's some wild and crazy stuff in here. Anyway, so the methods for analyzing the bacteria that we collected were threefold. One was we plated the bacteria on nutrient agar plates. Specifically, we did 3 plates, and this is where I'm a little less experienced. The TSA plates, or TSA plates and MSA plates and EMB plates. Each of these have a nutrient agar in them that selects for different kinds of bacteria. The TSA plate is considered sort of a catch-all. If there's bacteria on the sample, it should grow in the TSA plate. On the other hand, the MSA plate is a saltier solution, and it has a pH-sensitive dye added to it that changes color when certain bacteria metabolize certain things. And it primarily serves as a way of detecting Staphylococcus in your samples. And then the bottom one here, the EMB plate. The EMB plate serves as a plate to find indicator bacteria, specifically fecal coliforms. And fecal coliforms are bacteria that indicate the presence of, you guessed it, feces. So we plated our samples across all of these plates. We let them incubate, and essentially we saw what grew. We also did genetic analysis on the samples. So on the TSA plates, the ones that if there's bacteria, it should grow. We washed those plates, and we had a genetic analysis of them done to determine the variety of bacteria that's growing on those plates. So the results are fairly straightforward. What we see on the catch-all plates, the TSA plates, is that as people continue to use the displays, you see here we've got our 7 weeks, and across the bottom, our x-axis, we've actually got the hours of estimated usage of those displays. So the graduate students in our lab recorded the amount of time that each of those displays were utilized on a weekly basis. And what we see is that the amount of bacteria that shows up on the headsets increases with display use. So it does appear that the bacteria that's growing on these displays, that's transferred to these displays, is probably coming from the faces of the users. And so one of the neat things here, is that week 1 was our control week, where we sanitized the display with a 70% ethanol solution and swabbed and plated, and nothing grew. So that appears to be an effective way of sanitizing the displays. But as hourly usage of the displays increased, you can see that the number of colony forming units, that means little groups of bacteria that grew on the plates after we swabbed the plates from the swabs we collected from the displays. One potentially disturbing thing about this is that these levels are higher than what's typically seen in most computing labs when you're looking at keyboards that you've swabbed. So more bacteria than you'd find on your typical open lab keyboard. We found similar growth on the MSA plates. Remember, these are the ones that are selective for kinds of Staphylococcus. And this also kind of indicates that most of the bacteria from the TSA plates was probably a staph display, or staph bacteria. We do have some good news, though. The good news is the EMB plates, the one indicative of fecal coliform bacteria, had no growth at all. So no evidence of fecal contamination on your displays. So that was a good thing. But we have to follow that up with a little bit of bad news as well. We tested the bacterial colonies that formed for antibiotic resistance. And there was a lot of antibiotic resistance. We tested them against several common antibiotics, erythromycin, penicillin, gentamicin, and let's see what that other one is, tetracycline. And what we found is that there was a lot of antibiotic resistance among the staph variant of bacteria. The only one that showed much effect against preventing the growth of these bacteria was gentamicin, but they were surprisingly resistant to antibiotics. Now, don't get too alarmed about this. Staphylococcus, that family of bacteria is wide, and there's a lot of different varieties, and chances are you have it living on your skin right now. For most people, it's not a terribly threatening bacteria, but if you're immunocompromised or you have a wound or something like that, it does pose a risk of turning into a more serious infection. Now, this is this is the genetic analysis. We found a huge variety of bacteria on the headsets. The good news here is most of them are pretty common. So a lot of these are just the flora that grows on your face anyway. So nothing too terribly surprising there. Now, there are a few limitations that I do want to bring up very, very early. Well, as not very early, this is the end of the talk, that I want to bring up and emphasize very much. And that's that this study addresses bacteria, not viruses. So there's a lot of concern right now with sanitation and hygiene, especially obviously because of the COVID-19 pandemic, which is caused by a virus. Now, this paper does not address viruses, and it shouldn't be taken as a reference for handling viral contamination. However, it is important to note that viruses and bacteria, these two pathogens, usually have similar methods of spreading and similar means of being transferred from one individual to the next. So take that information as you will. So the 70% ethanol solution we used was effective against bacteria, but further research actually needs to be done to show whether or not similar sanitation procedures will be effective against bacteria, against viruses. Another limitation is we tested this using the factory faceplate, or not using the factory faceplate. We used an aftermarket sweat-proof faceplate that is hypothetically easier for us to clean. Our suspicion is that the factory ones are probably going to be worse since they're much more porous and sort of almost have a structure like a kitchen sponge. Another potential limitation is that our study was conducted in an uncontrolled environment. This was an open lab. Students, almost everyone who came in were either students doing research in the lab or students working on their projects for the class. But occasionally people would bring their friends or their roommates in to show their projects to. So it was an uncontrolled lab setting, which is why earlier in the talk on the slide, I wrote that was roughly 30 students. So it's 30 students plus, probably friends, roommates coming in and seeing their projects as well. But on the upside here, doing it in this kind of setting is probably more reflective of what VR practitioners will be experiencing in the wild. So some of the takeaway things here: VR headsets can collect and harbor bacteria. That's important. We got the numbers now to actually like see this. And the levels of contamination increase as the hours of usage increase. And Staphylococcus aureus was one of the primary, matter of fact, that was the largest largest group of bacteria that we found on the headsets. And this one is one that can potentially lead to infections in wounds or immunocompromised people. Some of the other bacteria were just, things that were common flora that grow on your skin. One of the really common ones we found was the bacteria that makes gym socks stinky. So if your VR display's got a funk, well, you know who to blame now. And the 70% ethanol solution appeared to be very effective in cleaning those displays. So we didn't see any bacterial growth after sanitizing the display with that solution. So that brings us to the end of our talk. And if you have any questions, send those guys over to Poll Everywhere, and I'm here to take them.
All right. Thanks for the talk, Adam. I think it was really interesting. And I think on the poll, a lot of people were really surprised and happy about the presentation. They have comments like, oh my God, I love your presentation. It's like watching a TED Talk. Yay. And it's so funny. And you were born to present. Happy face. So yeah.
Yeah.
Okay, but there's actually two questions like more serious.
Yeah. We need to stick to the topic. Flatter me on Discord. We can talk about the science right now.
Okay. So one is, you might have just written out public VR demos for me. Thanks, Adam. But do you have any data about whether disposable masks will prevent bacterial contamination?
So we didn't study masks. However, one of the things that since we did this study, looking at our early results, one of the steps that we added to our in-lab procedures was that people needed to have little donut-shaped masks when they put on their headset. Now, unfortunately, COVID happened. And one of the things we wanted to do was actually do this study again with our enhanced procedures and see how that turned out. Unfortunately, and it's actually kind of ironic because of the potential of contamination. We had to stop using the displays in the middle of a semester and start doing this all remotely. So I don't have any evidence on that. If I were to speculate, I would say that any separation you put between you and a potential contaminant is probably going to be a good thing. So masks, I can't say that they're effective for VR headsets, but it's likely that that's the case.
All right, I think I have like time for a really fast question. So
Okay.
70% ethanol was effective, like good to know, but does it not spoil the material over time of the cover?
Well, that's one thing that we don't know. So, I mean, it is a solvent, and solvents react with other things. So it's likely, I would suspect that it's likely that the plastic probably will break down more quickly with use and over time using an ethanol solution. However, these faceplates aren't that expensive. This might just be a cost that we need to build in to the overhead of running a virtual reality space in order to keep the users safe. So yes, it probably will break down the display, or the not the display itself, but the facial padding, because those are somewhat delicate, more quickly. But it's probably worth it in the long run.
All right, so I think we're run out of time, but there are more questions for you on the Discord. So please go and answer them when you
Awesome.
have time.
Thanks.
Yeah, thanks.